In this section, we are going to discuss the advantage and application of polymerase chain reaction. But let's discuss the question I have asked from you. And that was, what decides the temperature for denaturation of deoxyribonucleic acid? Well, we know that guanine has a bond with cytosine and adenine with thymine. But there is a difference in the number of hydrogen bonds between them. Well, guanine is bonded with cytosine through 3 hydrogen bonds, but adenine and thymine has 2 hydrogen bonds. So, as a result of which, more heat is required for the deoxyribonucleic acid having more guanine and cytosine bonds, and less heat for the deoxyribonucleic acid with more adenine and thymine bonds. Now proceeding towards our topic, let's discuss the advantage of polymerase chain reaction first. The major advantages of polymerase chain reaction over biologic cloning as a mechanism for amplifying a specific deoxyribonucleic acid sequence are sensitivity and speed. Deoxyribonucleic acid sequences present in only trace amounts can be amplified to become the predominant sequence. Polymerase chain reaction is so sensitive that deoxyribonucleic acid sequences present in an individual cell can be amplified and studied. Isolating and amplifying a specific deoxyribonucleic acid sequence by polymerase chain reaction is faster and less technically difficult than traditional cloning methods that utilize recombinant deoxyribonucleic acid techniques. Now moving towards applications of polymerase chain reaction. Applications of polymerase chain reaction has become a very common tool in research, forensics, and clinical diagnostics. First application is that comparison of a normal gene to its mutant form. So, polymerase chain reaction allows the synthesis of mutant deoxyribonucleic acid in sufficient quantities for a sequencing protocol without laborious biologic cloning of the deoxyribonucleic acid. Moving towards second application of polymerase chain reaction, and that is forensic analysis of DNA samples. Well, deoxyribonucleic acid fingerprinting by means of polymerase chain reaction has revolutionized the analysis of evidence from crime scenes. The deoxyribonucleic acid markers analyzed for such fingerprinting are most commonly a type of polymorphism, known as short tandem repeats. Deoxyribonucleic acid isolated from a single human hair, a tiny spot of blood, or a sample of semen is sufficient to determine whether the sample comes from a specific individual. Moving towards third application of polymerase chain reaction, and that is detection of low abundance nucleic acid sequences. Viruses that have a long latency period, such as human immunodeficiency virus, are difficult to detect at the early stage of infection using conventional methods. Polymerase chain reaction offers a rapid and sensitive method for detecting viral deoxyribonucleic acid sequences, even when only a small proportion of cells harbors the virus. Quantitative polymerase chain reaction, also known as real-time polymerase chain reaction, allows quantification of the amount. That means copy number of the target nucleic acid after each cycle of amplification that is in real time, rather than at the end, and is useful in determining viral load. Moving towards fourth application of polymerase chain reaction, and that is prenatal diagnosis and carrier detection of cystic fibrosis. Cystic fibrosis is an autosomal recessive genetic disease resulting from mutations in the gene for the cystic fibrosis transmembrane conductance regulator protein. The most common mutation is a 3-base deletion. That results in the loss of a phenylalanine residue from the cystic fibrosis transmembrane conductance regulator protein. Because the mutant allele is 3 bases shorter than the normal allele, it is possible to distinguish them from each other by the size of the polymerase chain reaction products obtained by amplifying that portion of the deoxyribonucleic acid. The results of such a polymerase chain reaction test can distinguish between homozygous normal, heterozygous, which is carrier, and homozygous mutant, who is affected individual. Moving towards next application, and that is polymerase chain reaction in comparative studies of genomes. The differences in the genomes of 2 organisms, such as bacteria and virus. So, the differences in the genomes can be measured by polymerase chain reaction with random primers. The products are separated by electrophoresis for comparative identification. 2 genomes from closely related organisms are expected to yield more similar bands. Moving towards next application, and that is polymerase chain reaction is very important in the study of evolutionary biology, more specifically referred to as phylogenetics, as a technique which can amplify even small quantities of deoxyribonucleic acid from any source: hair, mummified tissues, bone, or any fossilized material. Polymerase chain reaction has revolutionized the studies in paleontology and archaeology. The movie Jurassic Park has created public awareness of the potential applications of polymerase chain reaction. Now, let's discuss the sources of deoxyribonucleic acid polymerase in the original technique of polymerase chain reaction. Clinal fragment of E. coli deoxyribonucleic acid polymerase was used. This enzyme gets denatured at higher temperature. Therefore, fresh enzyme had to be added for each cycle. A breakthrough occurred with the introduction of Taq deoxyribonucleic acid polymerase from thermophilic bacterium, called as Thermus aquaticus. The Taq deoxyribonucleic acid polymerase is heat resistant, so it is not necessary to freshly add this enzyme for each cycle of polymerase chain reaction. The replication or polymerization of deoxyribonucleic acid requires a deoxyribonucleic acid polymerase enzyme. In polymerase chain reaction, Taq polymerase is used. It is thermostable and is isolated from a heat-tolerant bacterium, Thermus aquaticus. A thermostable polymerase is required so that it can remain active at higher temperatures. Higher temperatures are used in the polymerase chain reaction process for denaturation of the deoxyribonucleic acid double helix repeatedly for multiple cycles. Taq polymerase is most active at around 70 degrees Celsius. Now let's discuss variations of polymerase chain reaction. The basic technique of polymerase chain reaction has been described. Being a versatile technique, polymerase chain reaction is modified as per the specific demands of the situation. Some of the variants of polymerase chain reaction are nested polymerase chain reaction, inverse polymerase chain reaction, anchored polymerase chain reaction, reverse transcription polymerase chain reaction, asymmetric polymerase chain reaction, real-time quantitative polymerase chain reaction, random amplified polymorphic deoxyribonucleic acid, amplified fragment length polymorphism, and rapid amplification of complementary deoxyribonucleic acid ends. It was all about this video on this topic. In our coming section, we will discuss about the reverse transcriptase and allele-specific polymerase chain reaction. But before ending this section of the video, let me ask a question from you that: what do you know about primers for polymerase chain reaction?
Explore our extensive library of over 1,800 video lectures to learn about a wide range of topics. Only on sqadia.com.